Hi, my name is Xiang Zhang. I am a PhD candidate in computer science at Dr. Chad Meyer's lab at the University of Minnesota. I am presenting my research on leveraging machine learning essentiality predictions and chemogenomic interactions to identify antifungal targets. In this video, I will introduce our research problem about the fungal pathogen Candida albicans and talk about our machine learning pipeline. In the end, I will summarize our results. Candida albicans is a fungus. It's a common member of the human gut flora, but it's also an opportunistic fungal pathogenic yeast that can even lead to deadly infections in humans, especially in immunocompromised people. So it's very important to understand the function of Candida albicans genes in order to develop more effective therapeutics. And one key question in this field is, what is the essentiality of every gene in C. albicans? If we take a look at one of the model microorganisms, which is the baker's yeast, that shares more than 4,000 orthologous genes with C. albicans, we would find that under standard laboratory conditions, there are about 20% of yeast genes deemed as essential. So probably we can expect a similar rate in C. albicans. But building a mutant library in C. albicans could be much harder as those genes are less tractable. As a result, we cannot easily conduct large-scale genetic studies in C. albicans. Despite that, our collaborators at the University of Toronto were able to extend a collection called GRACE Library, which includes 2,327 conditional mutants, from which they identified 523 essential genes and about 1,800 non-essential genes. This is a wonderful ongoing project with years of hard work, but we can see that it's still less than half of the total number of C. albicans genes. So a very big motivation for this project is to drive the wet lab experiments to find more essential genes in a more efficient way. We would like to address this problem by reinterpreting the question as, can we build some machine learning model to predict the essentiality of the untested genes and make recommendations for the wet lab experiments? As described before, we have the GRACE collection of 2,327 genes, which were marked as essential or non-essential. Then, we also collected multiple features from both our collaborators and the published work, covering more than 6,000 C. albicans genes. We established a random forest-based machine learning model, which was trained and optimized on the GRACE standard and can predict gene essentiality for the majority of genes on the C. albicans genome. Then, we can recommend the predicted essential genes to our collaborators and have them prioritize constructing the mutants for those genes, which results in an additional GRACE V2 collection. Now, I'll introduce how we train, optimize, and test the random forest model. First, we split the GRACE library into 80% training set and 20% testing set. Then, we train our random forest classifier on the 80% training set with 5-fold cross-validation for hyperparameter tuning. After the optimization, we tested the performance on the remaining 20%. With a stringent cutoff of 0.5 for the prediction scores, we estimated an average precision of 0.77 and an area under our C-curve of 0.92. After that, we retrained our random forest model on the whole GRACE gene set using the optimal hyperparameters, and we got the permutation feature importance for every single feature. Finally, we predicted the essentiality for 6,638 genes. To assess the accuracy of our prediction model, we selected 866 genes for experimental validation, covering the range of prediction scores. We discovered essential genes at a rate of about 64% among these new predictions, relative to an unexpected background rate of essentiality of about 20%. We can also see that our predictions are able to discriminate the essential genes based on the distribution density of the prediction scores for the validation candidates. And here are the summary statistics for the predictions from our optimal random forest model on the GRACE V2 set. We achieved an average precision of 0.66 and an AUC of 0.95. The GRACE V2 collection is like a new held-out set and supports the performance and generalizability of our model. We finally confirmed 621 essential genes in total. We took them through some public databases and found that 149 of them do not have human orthologs, which should be really interesting potential antifungal drug targets. On the other hand, we paired our findings with drug targets to identify 97 compounds targeting 9 of our experimentally confirmed or predicted essential genes. 3 of them lack human orthologs and should be worth more attention in the future. In summary, we built a generalizable machine learning model to predict gene essentiality of the majority of C. albicans genome. Given these strong results, we helped facilitate the experimental validation of new essential genes and identify potential novel antifungal drug targets. At the end of my presentation, I would like to highlight that the results of our project got published in November 2021. For more details, you can refer to this paper or the PDF file of my poster. That's all for my presentation of the poster. I also want to thank my lab colleagues and all the collaborators that support this project. Thanks a lot for listening, and please feel free to ask any question via the portal platform or email.